Hello, this is Halamali, a bioengineering junior student at North Carolina Agricultural and Technical State University, working under Dr. Yoon, and this is Brain Microphysiological Systems using microfluidic devices to model Alzheimer's disease under mechanical injury. A little self-introduction. I'm a third-year undergraduate in bioengineering. My career goal is to pursue medicine. I'm not sure what specific field just yet, but I know I want to start medical school. Right now, I am immersed in research and clinical exposure. I'm a part of Dr. Yoon's neuroengineering lab. I also did a joint program with UNC Chapel Hill in nephrology, and I work at an urgent care. The objective of this lab is to engineer mini brains that emulate a human brain, allowing for an in-depth study of brain diseases, Alzheimer's, and their mechanisms. We take patient stem cells and culture and generate organoids that we later embed into a hydrogel and then into a microfluidic device, ultimately creating a vascularized organ that we monitor and observe. What is Alzheimer's disease? Alzheimer's disease is a progressive neurodegenerative disorder characterized by memory loss, cognitive decline, behavioral changes, and more. It has two main pathological hallmarks: accumulation of amyloid beta plaques and neurofibrillary tangles. Diagnosis includes clinical assessments, neuroimaging, and biomarkers such as amyloid beta and other pathology. Anti-amyloid beta antibody-based immunotherapy has emerged as disease-modifying treatment targeting the amyloid beta plaques. However, they came with side effects such as amyloid-related imaging abnormalities that manifest as edema or hemorrhaging. These amyloid beta plaques are extracellular deposits composed of primarily aggregated amyloid beta peptides that interfere with synaptic activity, inhibit long-term potentiation, and disrupts neurotransmitter release and receptor function. This leads to cognitive impairment and cognitive decline. These MRI scans illustrate Alzheimer's disease's effect on the human brain. Up here we have maps of 3 healthy controls versus 3 participants with Alzheimer's disease. These R2 star maps analyze iron concentration accumulation in the basal ganglia, and this is directly associated with amyloid beta pathology, further portraying a loss of symmetry and volume in the area as the structure thins, as well as deformation of its boundaries. This is seen right here, and again right here, and in this one as well. Mechanical injury accelerates Alzheimer's disease. So mechanical injuries are brain injuries caused by sudden external mechanical force. They disrupt the signaling pathways, which leads to altered neuronal excitability, ionic imbalances, excitotoxicity, oxidative stress, and more. It also impacts the yes-associated proteins and transcriptional co-activator with PDZ binding motif. These are transcriptional co-activators in the hippo signaling pathway that are responsible for regulation of cellular mechanotransduction, repair, and regeneration. This dysregulation of YAPTAZ can manifest in 2 ways. Number 1, hyperactivation, where these co-activators translocate excessively to the nucleus, leading to overexpression, or 2, suppression, where the activity is abnormally low, leading to insufficient activation of genes critical for regeneration and tissue maintenance. These mechanical injuries induce rapid accumulation of amyloid beta in the brain, which increases the risk of developing Alzheimer's disease. Hydrogel. Our hydrogel resembles and mimics the extracellular matrix of the brain, offering a supportive environment for the spheroid. The alginate and polyacrylamide crosslinks allow for easy cell encapsulation upon seating. We use a rheometer to measure the elasticity, the storage modulus, and the viscosity, the loss modulus of the hydrogel. But to limit cytotoxicity, UV light polymerization is avoided in the experiment. And I've also included the components we use in synthesizing hydrogel. Mechanical properties of hydrogel. This graph illustrates the differences between the loss modulus and the storage modulus of the UV polymerized sample versus the non-UV polymerized sample. Again, storage or Young's modulus measures the material's ability to store energy elastically, while the loss modulus measures the material's ability to dissipate energy through heat due to its viscous flow. The results indicated that UV exposure heightened the efficiency of the hydrogels. However, it creates a toxic environment for the cells, therefore it cannot be used in experiments. A common trend also seen in the graph is that viscosity decreases with decreasing angular velocity, while elasticity remains fairly constant. Microfluidic devices, aka organs on a chip. For microfluidic devices, we use a wafer that acts as a mold for our polydimethylsiloxane-based elastomer. This wafer is developed through a soft photolithography process. And down here, I have an enlarged image of the intricate design of our wafer, where we have 2 inlet chambers for cell suspension right here, and they connect into a perfusion channel. We also have 1 air chamber for stress application, in that trauma is mimicked through application of pressure on the membrane. Process. Ren cells, or human neural stem cells, are first cultured in a T75 flask. Upon desired confluency, we typically go for about 90%. The cells are transferred into a non-treated well plate where they begin to form spheroids. These spheroids are maintained for at least 3+ weeks and then integrated in a hydrogel. By this step, our microfluidic devices are already assembled, so the hydrogel encapsulated spheroids are then to be loaded into the microfluidic devices via the inlet chambers. Mechanical stress can then be facilitated, mimicking trauma to the brain. The cellular response is then monitored, and the spheroid can be stained for observance. Over here, we have images pre-mechanical trauma to the spheroids. Right here, we have a D4 neural spheroid under GFP, and these spheroids come with the gene of green fluorescent proteins, which means that they emit green under blue or UV light, allowing us to visualize it. Right here, we have 2 neural cell spheroids in a microfluidic device, and they're suspended in collagen, which is used as the extracellular matrix in this case. These 2 images are post-traumatic stress application. On the left, we have a histological section of a G10 healthy neural spheroid. It's also under GFP. And then on the right, we have that same G10 spheroid stained for YAP and DAPI in 10x. And we can visualize the cell structure and response via the glowing areas present on the cell. Histology. Histology is the study of microscopic structures of tissues and cells. Staining the cells prior to sectioning allows for the visualization and quantification of cellular signaling and structure. Mechanical stress application. So in an attempt to mimic brain injury, we subject the spheroids to controlled mechanical stimulation by deflecting the membrane within its microfluidic chip using precisely regulated pressures applied in a sinusoidal wave pattern. To achieve this, we use the Flow microfluidics controller, which is operated via a software. This controller utilizes two integrated pumps, one which generates positive pressure and the other which generates negative pressure, to create dynamic pressure differentials. Throughout the experiment, the chip is positioned under a microscope, enabling real-time visualization of cellular responses during the mechanical stimulation process. Discussion. Neural spheroids replicate the 3D architecture of brain tissue, while the membrane of microfluidic devices stimulate the brain's vascular system and fluid flow. As the pressure against the membranes increases, the deflection of the membrane in micrometers increases. This is visualized under this graph right here, which depicts deflection over pressure. And as seen on the graph, there is a positive direct correlation between the two variables. Membrane deflection refers to the deformation or displacement of a thin, flexible membrane in response to external forces. Down here, we have two images of the same spheroid before deformation in a microfluidic device, and then after deformation in the microfluidic device, following mechanical stress application. Conclusion. In conclusion, this design enables real-time observation of cellular responses, which is significant in understanding initial signaling mechanisms within the cells. Further analysis of the neural organoid is completed through histology, which assesses structural and tissue integrity, evaluates cell viability, visualizes cellular responses, monitors regeneration/repair. And this is all achieved through the staining of the spheroid for desired signaling pathways or responses. Future advancements include personalized patient models to produce personalized traumatic brain injury/Alzheimer disease therapy. And this concludes the end of the presentation. Thank you all.